of introducing our guest this evening, Dr. Mark Hunter, who is the Director of Women's Cancer. I'm sorry, yes,
That's
Women's
good.
Cancer at Ellis Fischel Cancer Center. And he'll be talking to us tonight about how looking into cancer research is giving us a little bit of insight into how aging happens and how maybe we can live forever, I hope.
Right, obviously.
All right, let's give it up for Mark.
Thank you. So we've been coming to the Science Cafe for probably about a year now, I guess. Sorry if I'm getting a little bit of echo. I'll move around a bit if I need to. So how many people in the room, I apologize for calling you out, how many people in the room are under the age of 30 right now? Okay, great. So I think that there's a pretty good chance that the people in here are under the age of 30, and I'm not going to say that 30 is some kind of magic number. I'm just going to go out and say, on record, there's a good chance that you can live to be 130, 140, 150. And the question will be, well, what are those ages going to look like? What's 122 going to look like? And in the case of the people who raised their hand, you're talking about 90 years from now. And the answer is 122, 90 years from now, is going to look profoundly different than it does now. But I don't want to start by talking about aging. I'm going to put those down for a second. Are you guys getting that ring, that weird echo, or is that just me? Okay. So let's talk about vampires instead. Can we take a minute and talk about vampires? Vampires is a cool topic. Vampires is a timely topic because it's still kind of Halloween, and we still have a lot of Halloween decorations up. So vampires is on the mind right now. Okay, so does anybody have any idea where the vampire legends first started creeping into kind of human folklore? So probably ancient Egypt. We're not totally sure, but there are some ancient Egyptian, like the Book of the Dead, described basically spirits that would roam around and usher people into the afterlife, and one of their sort of side gigs was draining the blood and the life force from the living. Why did they do that? We're not really sure. Somehow that life force ended up being something that would lead to immortality for the person that they were ushering into the afterlife. Not really quite sure how that kind of played itself out. And then, has anybody ever heard of Vlad the Impaler? So fast forward, millennia, and we come to this Vlad the Impaler subject. So Vlad was actually a prince. He actually probably wasn't as bad as everybody says he was. He was called Vlad the Impaler, of course, so that's a little bit weird. You're going to do some adjustments for us? So, who was Vlad the Impaler? Well, Vlad the Impaler was actually Vlad III, and he was a Romanian prince. His dad was Vlad II. Vlad II was knighted by one of the kings of Hungary, and in that knighting, he was given a surname called Dracul. Sound familiar? So, Dracul then had a son, Vlad III, and Vlad III was then Dracula, which means the son of Dracul. So that's where we get the term Dracula. Now, who was Vlad the Impaler? Well, Vlad the Impaler was just kind of this local guy that there was a lot of blood feuds happening and trying to recapture the throne of his dad and things like that. So what did he do? Well, he was pretty vicious. When he regained the throne, he invited all of the noblemen from this region where he was essentially the king and threw a huge banquet and impaled all of them. So that was kind of interesting. And so if the idea, I assume, spread fairly quickly, that if you're invited to a banquet at Vlad's house, you politely decline, you're out of town that day. And that's basically what happened with Vlad. So he was also known for some other unscrupulous behavior. So what does that have to do with anti-aging then? So we've got our vampires who drink blood, and we have anti-aging, and how are the two of them connected? All right, well, flash forward. Flash forward to 1955, 1957. So now we've got these crazy, really mad scientists. And if you're an animal proponent, you're a part of PETA, things like that, you may not like the concept of these experiments, but they are pervasive and they are providing us some information. And what those are, they're called parabiotics. Has anybody ever heard of the concept of parabiotics? A couple of people. So what that means is that we sew mice together. I know it doesn't sound great. It's really like the human centipede kind of thing. I've never seen that movie, but I know the general story of it, where they sew people together. It sounds pretty awful. We do it to mice, and we've been doing it for 60 years. But we sew them side to side as opposed to front to back like the centipede. So why do we do that? They share circulation. They actually create these anastomoses in the circulation where they can share circulatory systems. And we've been doing it to study obesity, and we've been doing it to study aging, and we've been doing it to study all kinds of maladies of the human condition, and in these cases, mice condition. So we sewed these mice together in the 1950s, and we discovered some interesting things. You can do what's called isochronic. Isochronic is really the chrono meaning time. Isochronic pairings or heterochronic pairings. So what's an isochronic pairing? You pair two young mice together, or you pair two old mice together. What's an heterochronic pairing? You pair an old and a young mouse together. And so what happens? They share circulatory systems. What happens to the old mouse? They become young. So that's some creepy scientific stuff right there. That's some creepy Halloween scientific stuff that basically shows that the concept of vampirism actually might work to restore youth. And how does it do that? We're not exactly sure, but we're beginning to study it. We're using these studies now 60 years later. Newer papers are coming out. They kind of abandoned this thing because it was gruesome, and we didn't have the ability to study it on a molecular, genetic, or epigenetic level, but now we do. Now we can take these pairs and figure out what's going on. Now we can figure out when you attach a young mouse to an old mouse, there are changes in the stem cell populations. There's a revving up. So basically what's happening in aging, among a great number of different things, is that the stem cells are dying out. They don't have the regenerative properties that they used to. And so what happens when so you would say, well, it sounds terrible that we're sewing mice together, and I agree, it's pretty awful. So what about just serum? Could we take the young serum and give it to the old? And what happens there? I mean, that's really the true form of vampirism. Vampires don't sneak in the middle of the night to your bedroom and sew themselves to your side. That would be way less cool than what they do, which is bite you in the neck. So what about serum? Can serum then cause that kind of regenerative rejuvenation of stem cells? And therefore, those stem cells, their ability to go ahead and repopulate the tissue, the local tissue? And the answer is yes. It appears that the replacement of old sera with young sera can do the same thing without having to attach them together. So that was a major find, and now what we're doing is we're trying, now that, as I mentioned, we've arrived at the ability to study the genes that turn on and off, this gene switches called the epigenetics or methylation or phosphorylation that turn on and off genes, and maybe even more importantly, the transcriptome. Does anybody know what a transcriptome is? Getting a little bit more common in terms. Transcriptome means all of the RNA really. So your DNA is transcribed into RNA. That RNA is transcribed into proteins. But RNA is not all transcribed into proteins. So really what we care about is the RNA. We care about its downstream transcribing, or how it's made into proteins. But because the RNA seems to be that sort of backbone by which everything else is run in the body, we're concentrating on that. And we can now take these mice, old mice, young mice, mice who've been attached, old mice attached to young mice attached to old mice, and we can run their entire RNA, every cell, of a particular tissue of interest, and what those cells are doing. And so that has begun to uncover facts about what it is that's causing, say, young sera to turn an old mouse young again, to turn on those stem cells again. We're being able to tweeze out and uncover those factors. And we'll talk about that in a few minutes. But before I talk about that, I wanted to talk about, and this is going to suck because you're eating. I'm going to actually grab my beer. Before I lay this one on you. Okay, so probably the most important and reliable and reproducible data we have on aging is related to the realm of nutrient deprivation. I'm going to say that as I take a sip of beer. Let people have a piece of their pizza. So what have we found over the last, well, I mean, it goes back to Warburg, studying the Warburg effect in cancer, but it goes back at least a century. What we have found is that if you deprive nutrients to an organism, that organism lives longer. How much longer? 50%. It depends on the organism, depends on the situation, depends on the level of deprivation. But from the smallest yeast, single-celled organism to the most complex organisms, and in this case, the most complex studied is macaw monkeys. So our closest to humans is the monkey population. Deprivation, nutrient deprivation, low calorie, and oftentimes low carbohydrate has been studied synonymously with low calorie, has been shown to extend life across the board. In 50, like I said, with as much as 50% increase in lifespan. They've been trying to study this stuff now in terms of not just lifespan, but they call it health span, because we want to know what the health span of an organism is based on certain, say, nutrient or genetic perturbations. We're less concerned about how long the organism lives, more concerned about how long it lives well, I guess is the way to describe that. Does that make sense? So now, again, in comes this RNA. In comes our ability to whole genome and whole epigenome-wide and whole RNA-ome-wide sequence these entire sets to determine what it is that nutrient deprivation is doing to organisms to cause them to live 50% longer. And there is a wealth of information coming out about that from every single pathway we can possibly look at those things in. We're trying to do this stuff because it turns out that there's thousands of genes that are differentially expressed in the starving versus the unstarving, or in the old versus the young. And so the difficulty is in how do you interpret that information, and that is becoming an important and new science. So how does this then relate to cancer? And that leads me to really the third. So if I were to describe these as three major areas of interest with regard to aging, the first, again, is the studies where they sew rats together. And the second is this concept of nutrient deprivation. The third would really be, as I have mentioned already, this RNA, our ability to do whole organism-wide sequencing of individual base pairs, really. And so I do want to bring up an interesting point, because we are talking about some crazy stuff, right? We're talking about finally reaching the point where our kind of organism can live for an extended period of time, and how impossible, or at least improbable, that is. But I want to put this into your head just as an aside. Does anybody know how many individual transistors are in the current iPhone 6? So a transistor is an on-off switch. Every computer that we have on Earth, well, for the most, I assume that's true. I think there may be some others, exist because it has the ability to turn on and off electric switches. It's either on or it's off. So how many switches are in an iPhone 6? Anybody have any idea? 6 billion is what I hear. Are you guessing? Okay, she's guessing. Anybody got another guess? 6 billion and one. So you've got to learn to play Price is Right better. You don't go, wait. You go under, right? You got to go under one.
You can do both. You can play
Is it over one? I've got to learn to play Price is Right better. It's been a long time since I was home at 10 a.m., unfortunately. So the answer is 3 billion. 3 billion. You were very good. You had the order of magnitude right, and that's the only thing that matters. How many base pairs are in human DNA? 3 billion. There's 3 billion base pairs in human DNA. There's 3 billion transistors in the iPhone. Therefore, we have arrived at the point where we can build a machine as complex as the human cell. So, is that absolutely true? Have we reached some kind of singularity? Is it all downhill from here? The answer is probably not. It's much more complex than that. It is a type of milestone for us. It really is. Because if I asked how many people had iPhones in this room, it'd be a lot of you. And we have the understanding to control every single element and aspect of that iPhone. We do not have the understanding to control every single element and aspect of ourselves yet. But you can see where our science is blazing forward in our ability to be able to do so. And I, again, contend that there are people in this room who will see the fruits of that within 90 years and extend their life significantly. Okay, so how does it relate to cancer? Well, cancer is an aging phenotype. I'm a cancer doctor, and most of my patients are, let's just say above the age of 30. I do have patients who get cancer who are young. That's just how cancer works. It's a series of genetic accidents, but they are genetic accidents that occurred for a reason. And what are those reasons? The answer is because the machinery was shutting down. So I told you the gatekeeper machinery has been shutting down. So I mentioned before that, that cancer is an accumulation of accidents, as is aging. So when we then looked at these young mice, old mice, whole RNA, the entire RNA world in an old subject versus a young subject, we can then divide the pathways. This pathway is related. So what are the pathways that are breaking down in the elderly? What are those genetic pathways or paragenetic pathways that are becoming dysfunctional in the elderly or as you age? And I want to say elderly, but there is no magic date. The truth is, it's just this clock that winds down. And so what are those pathways? There's dozens of them, probably 57. There are several that are particularly important. And what do they relate to? Inflammation is upregulated as you age. The immune response to either endogenous or exogenous insults is amplified as you age. Insulin and insulin balance and insulin homeostasis is perturbed as you age so that your system is bathed with more and more insulin, which is why people get diabetic as they get older. It's not just their diet, although that has a lot to do with it. It's also the aging process. There are lots of other pathways, several that lead to cancer phenotype. What are those pathways? You may have heard of the mTOR pathway. You may have heard of PARP inhibition. These are common words that we're looking at in cancer that basically control cell signaling. They control gatekeeper functions in turning on and off defects or killing cells when defects are detected. And all of that machinery gets worse as time goes on. So what are the relationships? That's exactly what we find, is that when that machinery becomes defunct as a result of age, cancer ensues. Does that make sense? So that is how aging relates to cancer, and that is how aging research relates to cancer research. We're very concerned with the same pathways. And I'll tell you that people care less about aging. They donate less money to aging. And so when you look at the whole of the literature, there is considerably less about aging as there is about cancer and how these genetic syndromes develop. But because there's so much crossover, we're learning more and more every day about aging as we continue to expand our knowledge about cancer. Does that make sense? So is everybody excited about living to be 150? No. Okay, again, it's add years. Wait, how does it go again? Quality to your years, not... No, forget it. I forgot it. Apparently, I need more of whatever it is. I need to go home and attack. Okay, so where do we go from here? How are we going to get this blood? At a minimum, the best we know is that serum in the young work. And so that's a real and super creepy thing. I hope you're all not. I hope there aren't any mad scientists in the room that are going to go home. What are the familial kind of consequences of having a grandma that lives to their side? I hope that there's some other way to do that. National implications of having a population that is now considerably more elderly than it is youthful. Walking around with you. Is it all bad? Is it a bad thing to take someone that has accumulated 80 years of knowledge, let's say in medicine or in engineering or in any other scientific or art discipline, and then give them another 80 years to then teach that information? The point is, there are some disastrous consequences of this coming science, but not all of it is disastrous. it is, so telomere or telomere length is basically a series of repeats that occur within the cell that wind down like a clock. We thought, because this was one of the very first things that we've sort of uncovered as the aging or biological clock, that it was going to be sort of a holy grail, like the baby mouse blood. I want to call it that. There's just no good way to describe that. But it turns out that it may be more of a, so again, with telomere length or with telomere length, the question is not whether or not, it's a question of causality. So that is to say that is telomere length a indication, like literally watching a clock tick down, if you change the clock, you don't change time. It's just an indicator of what the current time is, and that's kind of what we think about telomere length. We don't understand the functionality of it, what it occurs, why it occurs, or how it occurs, but what we are discovering is that it probably doesn't play as important a role, at least from a causative or from a biochemical standpoint. It, however, does function as a clock. And so you can use telomere length, and usually they use white blood cell telomeres or leukocyte telomeres, and they can use that to predict sort of the effects of aging. And so one of those things which I was going to have you go, kind of through an example of, but I've sort of talked through it, which is usually what I do, is meditation. If I was good at meditation, I'm not good. I've never been a meditator, but if I could have, I would have walked you through a short meditation, and I would have shortened your telomere length. So, when you go home tonight, meditate and shorten your telomere length. So how does it do that? It has to do with stress, and when you are involved in practices that reduce your stress and increase mindfulness, there is something to do with that mind-body connection that is real and biochemical and can be measured to the point where meditation can have a measurable effect on your telomere length, or in other words, your biological age. Does that make sense? So the telomere question is still hinging there, but this flood of RNA-seq data coming in regarding aging has kind of supplanted a lot of research in the telomere stuff. One over here. Yeah.
Imply that meditation shortens your lifespan, or is it?
So meditation, sorry, if I misspoke it, lengthens telomere length based on a UCLA study and a couple of other ones. So correct, it's supposed to increase your biological lifespan, decrease your biological age. Biological. Yeah, I think it's called biological. I'm not sure. I mean, is it chronological? Chronological is the time you were born. Biologic is what your organism looks like, and the two are not necessarily in complete sync. Nor are the individual cells in your body in complete sync with the organism as a whole. So again, we're tweezing out the mouse baby blood and what the components are, and why it is that the old serum can suppress and make a young mouse become older considerably in real time. We're not sure what that is or what those factors are. There are several of those factors that we're studying, and the MHC, I don't know if you guys are familiar with the major histocompatibility complex. MHC determines your blood type. So for some reason, the MHC B2 rises as you get older, and that may be one of the systemic hormonal factors found in the older mouse blood, if you will. Right. Are there any diets? Well, okay, so one of the questions is, is there a dietary plan that would change methylation? I don't know if it's that simple. They're starting to tweeze that out now. So so if you put patients on mice, let's say, in cancer studies, if you put mice on a low carbohydrate, high fat diet, they take on what's called a ketogenic phenotype, meaning they will make ketones in their blood. Those ketones are anti-cancer, anti-proliferative, anti-aging. So it appears that the best data we have with regard to diet and changing lifespan is related to high-fat, low carbohydrate diets. And in cancer, those same diets have been shown to significantly shrink tumor volume and metastases. And for that reason, I have put my patients on similar diets, and they've done very well on it. But the human studies at this point are lacking, and especially the connection to the methylation and phosphorylation, or otherwise the epigenetics associated with it. Again, aging ticks away at those pathways as well. And so there are connections on the level of sugar and fatty acid metabolism. Your sugar and fatty acid metabolism break down as you get older as well, which may be why those diets become more and more important in maintaining youth and decreasing the risk of cancer as you get older. So that is a really, it's a super important question. You can see that, China, which we thought was an enormous powerhouse of really production and economic, really a stranglehold on the rest of the world. Well, their economy's collapsing. And why is their economy collapsing? Because they don't have enough young people to take care of all the old people. It's already, their practice, which seems to make so much sense, which is reduce the population some way. And the only way that it seems to make sense to reduce that population is with birth control. And that can be a big problem. So what are we going to do? Who's going to take care of the elderly as the young population dies out? And it's occurring naturally anyway. We are, as a species, beginning to suppress our birth rate on our own because we realize how much of a problem that is for the planet.
So when you replace serum in the mice to get the rejuvenation effect, is that like a total replacement?
We don't know, but there was a recent paper that came. There's been a few papers, and there's been some data that has been suppressed, but and then republished. It's like a weird thing going on, and I'm not. It's early enough to where we need several more studies to tweeze this stuff out. But it appears that the effect is across all three germ lines, across all three germ layers. So if you're an embryologist, or even if you just had regular biology, you'll remember there's ectoderm, mesoderm, endoderm that kind of gives rise to all of the tissue in your body. And we've had some studies that have suggested that that these heterogeneous pairings have had effects on all three layers, therefore every layer of tissue in the body. And most notably, that's been studied in the brain, in the liver, because those seem to be associated most with aging and the problems associated with aging. But in the brain specifically, they've done some very, very elegant studies to show that the stem cells in the brain that give rise to neuroplasticity. We used to talk and teach how the brain doesn't regenerate. You don't get nerve cells don't regrow. Well, that's completely incorrect. Nerve cells are growing all the time, and they're dependent on the stem cell population. And it appears that these rejuvenatory young serum actually does stimulate those neurological stem cells to create new neurons in the older population.
So to get the effect on the mice, do they have to, is it a one-time shot, or do I need to drain her and then drain her?
Well, yes. Yes.
Because I'm really rich and I can afford to...
Right. You'll recognize that most of the vampires, are really rich, which happens when you live for 500 or 600 years. Now, for me, like if I live 500 or 600, I'm bad with money. And so I feel like if I live 500 or 600 years, I'll just be 5 or 6 times more debt than I'm in now. I kind of feel that's the way it would go with me. But
Your card appreciates you.
they very much do. But you can, the general vampire lore is that they do have to go from person to person. And I'll tell you, I've seen some studies that have shown monthly injections to try to maintain this kind of stuff. So if you just find a good, victim, I guess, once a month, they have to match your serotype, your MHC. B, you want to make sure that they're not, they don't have AIDS and stuff, because that's an issue that vampires really don't apparently consider very often. So you got to have some way of quick detection of those kinds of things. Yeah, both really good questions. So I'm going to tackle the first one second, and then I'll forget about the first one, and you'll just have to remind me. Because I haven't been drinking mouse baby blood. Let's just clarify that. So with regard to oh, I forgot both questions already. Let's go back to the first one. Is there 1,500 versus 2,000 calories? We're not entirely clear. The truth is that because everybody has different pathways with regard to gluconeogenesis and the glucose metabolism that occurs in the liver and in your cells, everyone is different, and everyone has a different dependency based on their level of exercise as well. I tell my patients 1,500 or less a day, but it's not a magic number. It's for simplicity's sake. I'll tell you that most of the studies have been around that range, but there's been studies that have also shown that fasting does exactly what we talked about. It's not just a even keel calorie restriction, but fasting can have an influence, which is to say that when you have complete calorie restriction, even if it's just for a day or so, you actually reset the mitochondria in your cells, and particularly those mitochondria in the white adipose tissue or white fat cells. You can fracture the mitochondria in the white cells, convert them to brown cells, and kick them into gear where they'll start spitting glucose out into your system and getting rid of it. So there is a growing body of science that would suggest that it may not be a strict 1,500 calorie a day thing, but there may be some benefit to an alternating fasting. And I've seen some studies that have looked at 1,500, 7, 15, 7, 15, 7, which is pretty drastic. Now, the question about exercise in the elderly, exercise in the aging population, that has been shown to alter those pathways. So if you are involved in exercise or start an exercise program as a patient who's older, it will invoke some of the youthful pathways that we are seeing in the young, and particularly those pathways involved in neuroregeneration. Yeah, from the standpoint of cancer research, stem cells is becoming more and more important. And what we're finding is that just like normal cells in the body, where you've got this stem cell population that's constantly rejuvenating and spitting out like your skin and your hair and all these cells constantly turning over, well, the same is true of cancer. There exists a subpopulation within a tumor that will continue to spit off little baby cancer cells that aren't necessarily stem cells. So there's these two populations, this kind of general population of tumor cells and this stem cell population of tumor cells. And the stem cell population is very interesting to us, particularly because it's resistant to chemo. So that is who are these stem cells? How are they resistant to chemo? What are the markers that differentiate and the pathways that differentiate them from the other cancer cells in the population? And yes, you're absolutely right. They are doing both exogenous, I guess you call it, and autologous stem cell transplantation. And we're not quite sure how that's working yet, but there are some studies that show the removal of cells from a person's body, converting those cells into a stem cell phenotype, in other words, biochemically making them behave as stem cells, and then putting them back in the body and getting those youthful, rejuvenatory cytokines or whatever the chemicals are that talk to the rest of the stem cells in the body, getting them all to rev up. So this is very, very early. Our understanding of the stem cells and what it means to be a stem cell and how to make cells become stem cells so that we don't have to be vampires. We can benefit from this stuff without being vampires, and the way we do that is we tweeze out the science. It is, again, it's an iPhone. We don't have to I guess, suck on mice babies for, a regular thing. There are biochemical ways around that. I guess probably the best way for me to answer that is to say that all therapy is quack therapy at some point. I mean, it all started that way, and if your grandma told you that the doctor came in 5 minutes ago, she's in Wisconsin, and you're on the phone, and grandma says, doctor said he's going to use leeches on my forearm because it's black, 30 years ago, you'd call that quackery, and today you'd say, well, that's part of our standard, medical therapy. Or the use of maggots to eat away dead tissue. Things that are quackery come back, or they're subsequently proven not to be. If you dig deeply enough into the literature, what you'll find is that, for instance, the Eastern philosophy and these thousands of herbs and naturopathic remedies that have been used by the Chinese and other ancient civilizations for millennia, they now have the research and financial apparatus to go back and study all this stuff. And what we're finding is profound. I mean, what we thought was quackery and really, from a Western standpoint, Eastern voodoo is quite scientifically sound when put to the test. And so I'm not sure, like, where the stem cell, yes, there are most definitely places you can go to get therapies that are not available in the United States yet. Those are for, safety issues, but there's also other political issues at play. And one of the studies I mentioned about the 3 ectoderms, they suppressed the 3, made them remove it from the paper. And I don't understand where all that political play was or why it occurred, but because it occurred, there was a 10-year lapse in the use of this type of technology in figuring out how to regenerate neural tissue. And so now they're looking at that data again and saying, what did we miss 10 years ago because it was suppressed, and what can we do now to try, in the United States and in the general scientific community to rev up that machinery? Anybody else? Well, thank you all. It was great talking to you. I really have a lot of fun talking about this. It's not my area of expertise. I really just, I primarily deal with cancer, but anytime, any scientific exploration, and certainly I can go around the room, and I have many, many times before on a Monday, and picked out unique and interesting people who are involved in unique and interesting work, and all that work goes together. I happen to have a gift where I can sort of see the forest from the trees from 10,000 feet, and everybody's important. Everybody's contributing. Everybody is doing something. What we're going to become, what we're going to turn into, who knows? But from my standpoint, it's a matter of suppressing human suffering. That's what I deal with on a day-to-day basis, human suffering in the form of cancer. And any work that we could do to try to fix that, even if it means that we end up dying from something else, I'm okay with that. Because cancer is a terrible way to die, and we want to do everything we can to fix that. So I appreciate everything that everyone in this room does in your contributions, but also in your work effort. But thank you for the evening. I appreciate talking to all of you.